In the last lecture, we looked in some detail at how DNA and cellular chemistry directs the creation of proteins in the cells and thus the creation of the whole organism. We talked about things like DNA replication, protein synthesis, mitosis, and meiosis. But when we looked at the inheritance, all that was said was that there were pairs of chromosomes and one member of each pair was inherited from each parent. This time, we'll look at how those different chromosomes interact with one another to create the organism's physical features. Our current knowledge of inheritance comes largely from those building on the work of Gregor Mendel, a 19th century Augustinian monk who conducted his work in the interval between Darwin's Origin of Species and Descent of Man. Mendel studied plant hybridization, that is, when two plants with different characteristics are cross-pollinated, what characteristics does the offspring plant have? His particular experiments were conducted on pea plants. This seems like a fairly simple research question, and Mendel didn't think of his research in any grand terms, but from a broader point of view, what Mendel was really doing was resolving the most serious flaw of Darwin's Origin of Species. You see, Darwin knew that favorable adaptations should be passed on to the next generation at a higher rate than unfavorable ones. To do that, there had to be some mechanism of inheritance, the process of passing traits to offspring. In 1859, when Darwin published, no one knew how inheritance worked. Most people believed it was a blending process, like pouring red mom liquid and blue dad liquid into a bowl, stirring it all up, and getting purple child liquid. But if that theory were correct, it would actually have been a huge problem for Darwin's theories. It would lead to the dilution of traits over many generations. Rare traits would vanish from the population rather than becoming more common. To see why this would be the case, let's think about giraffes' necks again. Lamarck had assumed that all giraffes started with short necks, but Darwin understood that there was always variation in that trait. At the start of the giraffe's evolutionary path, most giraffes would have short necks, but a rare few would have long necks. If only a few giraffes had long necks, then most of the short-necked giraffes would mate with other short-necked giraffes. Blending a short neck with a short neck would yield another short neck. Likewise, since long necks were so rare, most of the long-necked giraffes would also mate with short-necked ones. Blending a long and a short neck would produce a medium-length neck in the next generation. Then in the second generation, short-necked and short-necked pairings still produce more short necks, but there are still more short-necked than medium-necked. The result is that the medium-length necks would mate with short necks, resulting in offspring with necks a bit shorter than medium length. If this process continued indefinitely, eventually all giraffes would have the same length necks, which would be basically the same length as most started with. The result would be like adding a single drop of black paint to a 10-gallon drum of white. After much mixing, it would just look white. The rare trait would be diluted out of existence. In other words, natural selection predicts rare but favorable traits will become common. On the other hand, blending inheritance predicts rare traits, favorable or not, will dilute and vanish. Logically, both theories cannot be true at the same time. Since natural selection was supported by a mountain of evidence, the only conclusion was that inheritance must not be a blending process. But what else could it be? No one knew. Darwin recognized that this was a major problem, but it wasn't until Mendel's work that anyone proposed an alternative to the blending theory. Unfortunately, for a variety of reasons, Mendel's work was largely ignored for about 40 years, until the early 20th century when scientists rediscovered it. Then, in the middle 20th century, they were combined with Darwin's theories and the DNA discoveries we covered last time, and modern evolutionary theory was born. But what did Mendel actually discover? Well, the text covers the particulars of his experiments, so I'll just look at his conclusions. First, his major discovery was that traits are determined by discrete units, not some continuous material that can be blended. Rather than a liquid that can be blended, inheritance is more like marbles. When you pour red and blue marbles into a bowl and stir, you still have red and blue marbles, not purple ones. Today, we call these units genes. Furthermore, he discovered the principle of segregation. Genes occur in pairs, and one member of each pair is inherited from each parent. So, to skip ahead of Mendel a bit and back to modern understanding, we have a chromosome, which is really just a long DNA chain. At a particular point on that chain is a DNA sequence that codes for a particular protein. This location is called a locus, the location of a gene on a chromosome. But at each locus, there may be more than one version of the gene, each of which codes for different versions of the same protein. These versions are called alleles, alternate forms of a gene. Since chromosomes are in pairs, one from each parent, each cell has two alleles at the same locus. If those alleles are the same, the cell is called homozygous, having the same allele at the same locus on both members of a pair of chromosomes. If the alleles are different, the cell is heterozygous, having different alleles at the same locus on each member of a pair of chromosomes. Furthermore, Mendel realized that members of a given pair were not equal partners in determining the trait. That is, even at this level, there was no blending per se. Instead, genes for each trait he studied had at least two alleles, which he called dominant and recessive. A dominant allele was one that expressed in heterozygotes. If it was present, it dominated the other allele, making that one recede into the background. The recessive allele could only determine a trait, that is, it could only be expressed, if the individual were homozygous and there were no dominant allele to take over. These terms, dominant and recessive, are actually unfortunate choices. Especially the term dominant carries with it the connotation of stronger, giving the sense that this version of the allele is perhaps more common or more adaptive or just better in some way, but that's not the case. We don't know what makes one allele dominant and another recessive, but we do know that the only consistent difference between the two variations is which expresses in a heterozygote. There are dominant alleles that are very bad for you or very rare, and there are recessive alleles that are extremely common. So don't fall into the trap of equating dominant with better. These characteristics describe the genotype, the genetic makeup of the individual. Since Mendel was only working with traits that had only two alleles, there were only three possible genotypes: dominant-dominant and recessive-recessive, both of them homozygous, and dominant-recessive, or heterozygous. But remember, the dominant alleles can dominate the recessive alleles, making them recede and not be expressed. So heterozygous individuals would only express the dominant trait, just like homozygous dominant individuals. So there were three genotypes that resulted in two physical characteristics, the dominant trait or the recessive trait. This is called the phenotype, the observable characteristics of an organism determined by its genotype. Here's an extended analogy to help you understand the relationship between genotype and phenotype. Think of your genetic makeup, your genome, like two bookshelves, each with 23 books. These two libraries are sets of cookbooks, one inherited from each parent. Each volume represents a chromosome. Each set has the same 23 volumes. Let's say that volume 12 is desserts. So you have two homologous chromosome number 12s. Each will contain recipes, that is, genes, for the same desserts, that is, proteins. The page on which a particular recipe is located is analogous to the genetic locus. The XYZ locus on chromosome 12 is analogous to page 57 of chromosome 12. There, in both copies of the volume 12, you'll find a recipe for chocolate cake, but since these are different editions, you might not find the same recipe. One night, I decided to make chocolate cake for dessert. I pull out both volume 12s and flip to page 57. Mom's cookbook gives me a recipe for German chocolate cake, but Dad's gives the recipe for devil's food cake. This is a heterozygous locus. I have two recipes, but I only need one cake. Which one do I make? Well, based on whatever criteria, I decide to make devil's food cake. This makes devil's food the dominant cake recipe and German chocolate cake the recessive version. But there are two other possibilities when I first look at page 57. If both cakes give two identical recipes for German chocolate cake, this is a homozygous locus. Since we know from the last scenario that German chocolate is recessive to devil's food, we call this a homozygous recessive locus. Whether it's recessive or dominant, though, I only have one recipe, so I can only make German chocolate cake. The same is true if I look at both books and find two identical recipes for devil's food cake. The locus is homozygous dominant. With only one recipe, I obviously bake devil's food cake. There's no choice. The same sorts of scenarios are playing out all the time in all of our cells. The cell needs to produce a protein, looks at the two alleles it has at the relevant locus, and then activates only one allele. The only time there's a choice is when the locus is heterozygous, and in those cases, the dominant allele activates. When it came time for a parent to have offspring, only one of those two alleles could be inherited by each child. In homozygotes, it didn't matter which allele passed on because they were both the same. However, Mendel realized that heterozygous individuals could pass on either the dominant or the recessive allele to their offspring, and which allele went to which offspring was a random chance. Today, we know this is due to random assortment, the random distribution of chromosomes to daughter cells during meiosis. Finally, one of Mendel's slip-ups, the principle of independent assortment, the idea that genes influencing different traits are inherited independently of one another. This is not really true in every case. During meiosis, whole chromosomes move from one mother cell into the gametes. So two alleles on the same chromosome would both be inherited, or neither one would, and offspring couldn't receive one allele and not the other. On the other hand, if the loci governing two different traits are located on two different chromosomes, those traits are inherited independently of one another because of random assortment. Mendel believed the principle of independent assortment applied in all cases because chromosomes were unknown in his day. They weren't discovered for another couple decades. By complete accident, when Mendel chose which traits he'd study in his pea plants, he chose traits that occurred on different chromosomes. So in his data, those traits were inherited independently. But had he chosen a different set of traits to track, he'd have seen something very different. Okay, so far we've talked about Mendelian inheritance, inheritance of traits that are governed by alleles at only one locus. To call a trait Mendelian thus describes its genotype. Mendelian traits are also called discrete because they create phenotypes that fall into only a few non-overlapping categories. The term discrete, though, describes the phenotype of the trait, not its genotype. However, all Mendelian traits are discrete and vice versa. Your book uses blood types as an example of a discrete trait. Either you are A, B, AB, or O. There is no middle ground between AB and O. There's no overlap between A and B. Your phenotype must belong in one and only one category. It's like sorting billiard balls into boxes. Each person is a billiard ball and each phenotype is a box. Every ball goes in one box, and once it's in a box, it can't also be in another at the same time. But of course, there are plenty of phenotypic traits that are not discrete, but rather continuous. Rather than non-overlapping categories, these phenotypes are measured on a continuous scale with middle ground and overlaps. These are polygenic traits because they are governed by a combination of alleles at many loci, and usually a variety of environmental and developmental factors too. Thus, polygenic describes the genotype of a trait, and continuous describes the phenotype, but the two terms can be used interchangeably. Height and hair color are polygenic traits. People can be short, tall, or anywhere in between. There is overlap and ambiguity in which label to apply to a particular person. The truth is, Scientists are only just beginning to understand the genetic underpinnings of polygenic traits, which happen to be the great majority of traits that a physical anthropologist would be interested in describing. Okay, so in Chapter 2, we covered the basics of Darwin's natural selection. In Chapter 3, we looked at the functions of DNA and cellular chemistry. In Chapter 4, we've looked at Mendel's work on inheritance. Let's put all three of those bodies of theory together to see what modern evolutionary theory looks like. Beginning in the 1930s, the earlier models began to come together. The modern synthesis was the bringing together of multiple lines of evidence to integrate Mendelian genetics and natural selection within evolutionary theory. When Watson, Crick, and Franklin unraveled DNA in 1953, it filled in gaps in the then modern synthesis, and today we can say that modern evolutionary theory relies on all three bodies of theory equally. If you prefer, you might say that this three-part combination is the modern synthesis. In this view, we might envision the modern synthesis of evolutionary theory as a three-legged stool. The seat of the stool is evolutionary theory as we understand it today. Each leg represents one of the three contributing bodies of theory: natural selection, Mendelian inheritance, and DNA. More often, textbooks will combine Mendel's work on inheritance and the 20th century work on DNA into one category, calling it genetics. But if you remove any one of those three legs, the stool falls over. All three bodies of theory are equally important for understanding evolution because each answers questions that the others do not. If we focus on the seed itself, we can see how those three bodies of theory combine. The modern synthesis views evolution as a two-stage process: the production and redistribution of different alleles within a species, followed by the action of natural selection on that variation. The first phase is when genetics is at work, creating variation by making new alleles and combining them in different ways to create different phenotypes. During the second phase, natural selection promotes some phenotypes over others. Remember that one of Darwin's insights was that variation had to exist naturally before natural selection could act on it. Natural selection does not create anything. It just promotes one version over another. So without genetics, we can't have natural selection. But before we can explain the changes brought about by each stage of the evolutionary process, we need to be able to define exactly what we're interested in. If evolution is change, what kind of change? The modern synthesis defines evolution in this way. Evolution is a change in allele frequency in the gene pool of a population from one generation to the next. Notice how this definition draws on all three parts of the modern synthesis. It talks about alleles, that is DNA, their frequencies in different generations, that is inheritance, and also about the population, which is part of the definition of natural selection. Evolution by this definition applies to the genotype. That is, suppose for a given locus in a given species, the allele frequency is 60% dominant to 40% recessive. That is, 60% of all of the alleles are dominant and 40% are recessive, but remember that each individual has two alleles, so fewer than 40% of the population will have the recessive phenotype. Let's say that 50% of the population is homozygous dominant and 30% are homozygous recessive and 20% are heterozygous. The phenotype ratios in the population would be 70% dominant, 30% recessive. Now, suppose that the next generation has a 50-50 allele ratio. 30% are homozygous dominant, 30% homozygous recessive, and 40% heterozygous. The phenotype ratios in the population would still be 70% dominant, 30% recessive. The phenotypes may not have changed, but the genotypes have. Evolution has occurred at this locus, even though no outward signs can be detected. That example used the concept of a population, which is an important concept in understanding the modern definition of evolution. A population is a group of organisms of the same species from which individuals in the group usually find mates. In other words, a population is an interbreeding group. All individuals of a single species can interbreed with others of the same species, but that doesn't necessarily mean that they necessarily do. Maybe a river or a mountain range separates them, or in human populations, maybe it's religious or cultural differences. Whatever the case, almost any species is divided into a variety of populations, often with somewhat fuzzy boundaries. It's the population that is the basic unit of evolution. Technically, species do not evolve. Populations do. Before we move on to the mechanisms that play roles in the modern synthesis, there's one last point to note on the definition of evolution. Since evolution is a change of allele frequencies, even without a change in phenotype ratios, it may take place without any outward signs. Since it can happen at a single locus without a change at any other locus, it may produce extremely small changes in phenotype. Small changes are unlikely to make two individuals unable to mate if they would otherwise have been able to do so, so some evolutionary changes take place without speciation. This is called microevolution, the evolution within a species. Since all evolution boils down to small changes from parent to child, all evolution is microevolution. What Darwin realized is that small incremental changes could accumulate over time to produce new species. Generation 2 might have evolved from generation 1, but they were still the same species. In fact, in general, parent and child will always be the same species. But generation X and generation X plus 10,000 might not be, because each generation introduces a small bit of change. This long-term accumulation creates new species and is called macroevolution. Macroevolution is a long-term consequence of microevolution. It is not a thing in and of itself. It's just what happens when you do microevolution over and over. Either both are true or neither is true. And we have an overwhelming mountain of evidence that microevolution happens. So macroevolution is also true. The last major topic of this lecture, then, is the different mechanisms of evolution. Natural selection, contrary to popular opinion, is not the only mechanism that changes allele frequencies. There are several other ways that a generation can differ from the one before, and natural selection is only one of them. Okay? These mechanisms all operate during that first phase of evolutionary change, when we're creating and redistributing variation that will later be acted upon by natural selection. The only mechanism of evolution that actually creates new alleles is mutation. This is what happens when the cellular chemistry of DNA screws up and the molecule does not replicate itself exactly. It can happen for many reasons, other chemicals in the cell, that is toxins, radiation, or simple statistical probability. Regardless, mutation is absolutely vital to evolution. Darwin presupposed the existence of variation in any population. Variation requires multiple alleles at a given locus, and mutation is the only way to create new alleles. Without mutation, evolution would not occur. The next mechanism is gene flow, exchange of genes between populations. Consider two populations, each with different gene pools. Since they're not completely reproductively isolated, occasionally an individual from population A will mate with an individual from population B. Their offspring will have genes from both populations. If he then mates, his children will belong to population A, but have genes that were not originally part of the population A's gene pool. Over time and successive interbreeding between the populations, genes that were common in population B but rare in A will spread into A's gene pool and vice versa. Since later generations have different allele frequencies than earlier generations, this is evolution, even if no natural selection has occurred. Next is genetic drift, evolutionary changes that are produced by random factors. The smaller a population is, the greater the effect of genetic drift, but since small is a relative term, genetic drift is possible in any population. When you flip a coin, there's a 50-50 chance of heads or tails, but it's possible to flip a coin 10 times and get 10 straight heads. Likewise, it's possible that a heterozygote with a dominant trait will have 10 offspring, all of whom inherit the recessive allele. It's also possible that they'll be homozygous and express the recessive trait. It's a truth of randomness that sometimes random processes appear not to be random. Next, there's the founder's effect, a special type of genetic drift, in which allele frequencies are altered in small populations that are taken from or remnants of larger populations. Suppose we have a population of people with a 50-50 allele frequency for some trait. It may be evolving or may not. Now suppose for completely non-biological reasons, a part of that population moves away. Maybe they belong to a religious minority and they're expelled from their homeland. They move to a new territory and found a new population. Also, suppose for whatever reason, the religious minority had a 75-25 allele frequency for that particular gene. The colonists' descendants in the new population will have a very different gene pool than their ancestors in the old, larger population. Finally, recombination can also play a role in evolution. Sometimes during meiosis, members of a chromosome pair will swap genes. This is called recombination. Recombination doesn't necessarily cause evolution in itself, since there are still the same number of genes, but they're distributed differently in different gametes. But by moving genes around, changing how they're combined in particular offspring, and putting them closer to different alleles, recombination can change how phenotypes form, and this can have an effect on the reproductive success of the organism. To summarize, mutation, gene flow, genetic drift, founder's effect, and recombination are all operating during the first phase of evolutionary change. They're all random processes. They can improve the gene pool by creating new phenotypes that are more adaptive, or they can harm the gene pool by creating new phenotypes that are less adaptive. All they do is create new variants. Finally, during the second phase of evolutionary process, natural selection chooses between those variants. Only natural selection is directional in that it consistently selects for the more adaptive traits. In wrapping this lecture up, let me say that I realize there's a lot of complicated material here. Read the text carefully and make good use of the discussion forums to clarify points that are still fuzzy. Let's go into our first exam strong.